Thank you so much. And thanks everyone for coming. Yeah, my name is Leisha. I'm a student at University of Queensland, currently undergoing my honours project. And the topic of my honours is kind of looking at microbial interactions in legumes and the dynamics of that. Um, but I guess to start, a little bit about me. I've been interested in mycology since I was a teenager. I imagine my interest started, like many of you, walking through the forest and seeing these little guys around and being curious about those. And as I got older, I started to get interested in viticology, actually, and maintaining vineyards and stuff. And Arbuscular mycorrhizal fungi are actually a large part of viticology, especially with the yeasts involved in fermentation and everything as well. Um, but yeah, after traveling for a bit, when COVID hit, I figured it was time to get into uni, and I really wanted to explore AMF and the beneficial properties of uh, fungal partnerships with plants, because I find that a lot of the fungal and mycological research kind of center around fungal pathogens. But I'm really interested in looking at the beneficial symbioses, which is what I've been doing. So I guess today I'm going to be starting with a bit of background on Pongamia pinnata, um, and why legumes are so interesting and valuable, why I chose them as a host, because we can't actually study AMF in isolation. It needs to have a host plant. So it took a long time to settle on Pongamia as my chosen host, but yeah, then we'll get into AMF and the methodology of preparing plant roots for microscopy and what structures you can expect to see there. So yeah, I guess we've got, oh hey, I guess we've got some current global issues that I'm hoping that my research addresses. With increasing global populations, food security is kind of becoming a growing concern, particularly with climate change and deforestation and land use changes. So, arable agricultural land, potential arable agricultural land being turned into other things, and agricultural practices, overuse of chemical fertilizers and pesticides, and even the fertilizers that we use today. Only 30%, around 30% of the fertilizers that are applied to agricultural fields are actually absorbed by the plants, and the rest is in runoff, which causes all of these domino effect issues and environmental concerns. But along with that, there's also an energy security issue, and the environmental implications of our mining industry. So I think there's a real important need to look for alternatives that are not just environmentally friendly, but also efficient and useful for real world applications. So this is where biofuel comes in. So biofuel's always been around. Wood is a perfect example. Creating fire, that is a biofuel. It's a renewable source of energy that can be stored and transported, which is what makes biofuel. Coal is obviously another one, but it's albeit less renewable considering its developmental time. And solar energy, although it is renewable, it still requires a battery that needs precious minerals that are not always gained sustainably or ethically. So there can be issues with that as well. But one of the benefits of a plant-based biofuel is that the emissions that you can get from burning that fuel is mostly just carbon, especially with a plant like Pongamia. The emissions that burn off is very low levels of carbon, but that carbon just gets fed back into photosynthesis as a part of like the carbon cycle and what makes plant-based biofuels so beneficial and efficient. Um, so Pongamia pinnata is a fantastic source of this biofuel. Not only for biodiesel, but it also has use in herbicides, insecticides. It's used in soap and other skincare products. Can be used as animal feed, but it's also a really pretty tree. There's almost 20,000 Pongamia trees planted all over Brisbane just because they have really nice flowers, they're easily maintained, they grow really easily. Um, but one of the huge things, which is what I'm interested in, is its potential as a biofuel crop, which is emerging in Australia. So it's originally from India, but it went through Southeast Asia and landed in the tippity top of Queensland. So it's a northern Queensland tree. So depending on who you ask, it can be either native or naturalized. But one of the major things in that it can grow in marginal soils. So it does not need fertile soil to thrive, which means it doesn't overlap with food agriculture and it doesn't take up land for that. So that's a huge benefit, and it can be planted essentially anywhere, which is great in Australia because we don't have a lot of arable land here. The other thing is that it has really high oils in that have steric and oleic acids, which is essentially the compounds you'll find in jet fuel, which makes it a really efficient, high energy biodiesel. Each tree can produce almost 100 kilos of seeds per annum, so it's fantastic high yield as a crop. But each of those seeds also have a really nice high oil content too, almost 40%. Um, and that's also a huge benefit for yield as well. But after you press that oil out of the seed, it also has a high protein content with the meal that's left behind, which is what is used in animal feed. So nothing is left behind. We're using all those parts as well. But yeah, as I mentioned before, it has a really low carbon emission burn off. So it's quite sustainable in that sense with a less greenhouse gas emission. And it's also really easy to harvest. You just shake the tree and the seeds fall out. It's like non-invasive, non-harmful, non-violent, really easy to do. But one of the main things is that it's a legume. And legumes are really, really unique plants. One of the great things about legumes is its ability to nodulate. So legumes are this is a nodulation that you'll see in plant roots in that picture that I've found. So they have the ability to develop these nodules, and that is inhabited by a soil-borne bacteria called rhizobia. And that rhizobia is just a nitrogen fixing bacteria. And that means it has this unique ability to transform atmospheric nitrogen into a plant soluble form, which is ammonia. And it's really, really fantastic. It's a high reward system because two of the most limiting resources in agricultural fertilizers is nitrogen and phosphorus. So it's essentially able to fertilize itself with this symbiotic partnership with the soil-borne bacteria. And that's what makes legumes so fantastic. Currently, some farmers will use legumes in intercropping with their crops. So whatever they're growing, they'll alternate the rows with a legume species to be able to reduce the amount of chemical fertilizer that they need to use because of this ability. But with high reward, it also comes with a high demand for resources, which is why it's really important for the plant to regulate how much of the symbiosis and infection of these microbes. So it's trade-off with the microbes, essentially giving the microbes carbon, and in return it gets sugars and nutrients. But carbon is also needed in the plant to grow as well. So it's a bit of a trade-off, and it needs to decide how much it's going to use that pathway essentially. So this is what the plant needs to decide with not just rhizobia, but AMF as well, because that is just another soil-borne symbiont that the plants can utilize. And one of that ways is if you don't, I'm sure you're all kind of familiar with AMF being in the Mycological Society, but arbuscular mycorrhizal fungi don't have fruiting bodies. That's what's known as the mycelium network in the ground, acting as root extension networks and increasing the plant's ability to absorb nutrients. Phosphorus is a huge part of that. So that also being the other limiting resource in agricultural fertilizers. So you can imagine the partnership between legumes who have this nitrogen fixing ability, and arbuscular mycorrhizal fungi that have a huge ability to increase phosphorus uptake. Like the combination of that has widespread implications in agriculture. But the great thing about arbuscular mycorrhizal fungi is that it's absolutely everywhere. It's present in almost 80% of terrestrial plants. I think there's very few plants that don't have it. I know the Brassica family doesn't form symbiosis with AMF. It's why I don't eat vegetables. Just kidding. But yeah, I think Arabidopsis, which is another plant model organism, it doesn't form symbiosis, but pretty much every plant that you'll see will have AMF in its roots. But yeah, it's got so many other benefits, increasing soil structure just in terms of the physical network that it's creating in the roots and then spreading out, and increased microbial activity in the rhizosphere. So it's been found that some bacteria will use AMF as a kind of highway to transfer from plant to plant as well. It's just got a lot of really cool, interesting things. But there's other major mycological benefits, and I think it's a really underutilized field of research for science because there is so much it can do, and it's such an important and intelligent organism. There's been studies in mycorrestoration, which is essentially creating more fresh soil. So it's also really good in climate change mitigation, as almost 80% of the world's carbon is in our soils, and the mycelium network is a big chunk of that. For every step you take, there's approximately 500 kilometers of this mycelium network beneath your feet. There was a study in 2020 in California that saw remediation of an abandoned logging plantation that had just gotten, it's just gravel. It was just gravel. So basically, what they did was they inoculated that land with a type of mycelium, and within 2 years, they got 5 centimeters of topsoil, which is huge. Normally, it can take 100 to 1,000 years, depending on the environment and the conditions, of course, to just generate 1 centimeter of topsoil. So it's obviously very underutilized, and we should be doing more here. But also microplastics are also kind of a huge concern, especially at the moment. It's very topical. I think the statistic is that we're eating one credit card's worth of plastic every year through microplastics consumed in our food and water. And different mushroom strains have been found to have this ability to decompose such toxic materials, including microplastics and heavy metals, even biodiesel waste and toxic waste. So there's also the use of microagriculture, which is kind of my area of interest, using fungi for pest control and fertilizer. For pest control, I'm sure all of you are familiar with cordyceps. That's probably the most popular pathogenic fungi, the zombie fungus. But yeah, as mentioned, like AMF being useful in reducing chemical fertilizer use, in increasing that nutrition uptake for the plants. But yeah, why don't we have more AMF research? There's lots of reasons for this. Research is lacking in areas like in AMF, in colonization, and its persistence in diverse soil types and agricultural practices. There's limited applications. While the potential for AMF is clear, the on-farm application is often limited, partly due to the lack of readily available effective AMF inoculants and the need for specific management practices. There's not a lot of understanding for AMF plant interactions, and more research is needed to understand the complex interaction between AMF and plants, including the molecular mechanisms involved in nutrient uptake and control and regulation of that infection. There's also not a lot of research done in diverse crop species. So research is mainly focused on a few crops and leaving gaps in our understanding for other plant species. And although AMF play a crucial role in soil health, more research is needed to fully understand their interactions with other soil microorganisms, not just the crop, and the impact of various agricultural practices on AMF communities. Um, so what we should be doing is more. It would be great if we had more funding, of course, and more interested scientists, of course. But yeah, it'd be great to focus research on identifying and developing AMF inoculants that are effective in various soil types and agricultural systems, being able to develop mechanisms and protocols for farmers that are easy to apply and, you know, learn and perform. Um, Encouraging crop rotation is a huge part of that. Reducing tillage and organic farming methods can also help support AMF populations and overall soil health. But yeah, let's just get into the AMF anatomy first, I think would be great. When you're looking at AMF under the microscope, there are a few structures that you can find. Spores is often on the outside of the root. Um, they are characterized by oval, they're usually quite dark as well, sometimes ornamented with different textures on the inside. Hyphae, which you might all be familiar with, you know, when you're going for a walk in the forest and you pull up a log and there's all that white matted mycelium network, that's essentially the hyphal threads, like filaments, and you can see that here. And these are actually vesicles. So the spherical, oval, bubble-like structures that kind of act as storage structures to store carbons and sugars in the trade-off. There's also arbuscules, which kind of look like little coral or tree-like structures that are able to transfer that material as well. So I thought it'd be fun to do like a little pop quiz to see what you think these structures are. So feel free to put it in the chat. So we'll start with these, what we think these ones are. Getting some good guesses in. Nice. Cool. Some good guesses kind of across the board. These are actually vesicles. So the spores are also a really good guess. Spores tend to be a little bit larger than the vesicles, a little bit darker, and tend to be a little bit more textured. I think the thing that you can find with vesicles is that they do look like bubbles. They're kind of transparent. They kind of don't look like they have anything in them. So that's kind of what's useful in identifying vesicles. Also, you can usually find hyphae attached to vesicles when you won't normally find hyphae attached to spores unless the spore has germinated and starting to like spread. But I think the clustering of these and the hyphae connecting them is a really good indication that they are vesicles. All right, we've got these ones. So you might notice like these structures here. Yeah, perfect. Everyone's pretty much nailed it. I think arbuscules are definitely one of the easier ones to identify because they just look like little trees in the cells and they pretty much take up the entire plant cell and you can find them pretty widespread across. Yeah, they're like little corals, so I think that's the easiest one to identify and they're super cute. Next one, we've got this. What do we think this one is? Yeah, nailing it. So yeah, perfect. So we've got some spores here. Perfect. This one you can see they're larger. The hyphae takes up much less space. So these ones have obviously germinated. We've got a little one here, so it might be early. But yeah, the textures in the spores are a great indication that that is what they are, because it holds all of the genetic material inside, and they are actually quite easy to see normally. So good job, everybody. This is the last one. Nice. Good guesses. Good guesses. But I actually tricked you. This is not even a plant. This is actually human hair. How crazy is that? Gotcha. Um, yeah, there's obviously a lot of similarities in microscopy. When you look at something closely enough, then they can always look like other stuff, but pretty cool. So you can find heaps of different structures. We got the vesicles here, hyphal clusters here. You can see how the more you look at it, the more it's kind of easy to identify. Here's a really good group of arbuscules here as well. You start noticing how characteristic some of these structures are, and they become kind of easy to spot. But that's if you have cleared everything well. That's if you've prepared the root samples well. And at the end of the day, to be perfectly honest, you can get 1,000 photos under the microscope, and only 5 of them will actually be good. And I'm looking at these, and they probably took thousands of photos to get a couple of these good ones. And you can see even this one, it's quite messy, difficult to see, and that is kind of what you might see a lot of the time. So the preparation in the plant roots are actually really, really important. And there is so many different variations in the kind of recipe that you might find in doing so. So this is the general protocol that you will follow, especially as a scientist when I'm trying to look for just AMF. Really hard to do in plants that you'll kind of pull from the wild because they'll have so many other microbes in there. There's fungal endophytes that can look really similar. Obviously, fungal pathogens, they're kind of everywhere. Um, but they might not, even in a healthy plant, it might not be showing signs of disease, but you might still find fungal pathogens in there. But that's why, as a scientist, when I want to make sure that all I'm finding is AMF, the seed sterilization is a big step. But if you just want to look at them under the microscope, then just go for your life. I usually do a chlorine gas sterilization, but you can also do ethanol sterilization with like 70% ethanol. You just douse the seeds and have them in there for literally 5 seconds before you wash it and just rinse it out with water like 10 times to make sure that there's zero traces of ethanol left. But then you can chuck them in and start germinating. Once they emerge, you transfer them to a pot and you inoculate with your inoculum of choice. Obviously, we're doing AMF. This diagram was taken from my research slides, so I've got the rhizobia in there as well. But yeah, and then you monitor for growth. The time that you need to wait for AMF to infect the plant can vary on conditions, environmental conditions, the type of plant, but also the type of AMF that you have. So strains can take different times to infect, and each of those strains might have a relationship with different plants as well. And it's also important to note that not all strains of AMF will infect all types of plants. So that is obviously another growing body of research as well. So I'm finding with soybean, it needs at least 6 weeks. For other types of beans, because I'm obviously looking at legumes, sometimes you can see it within 3 weeks. From Pongamia, I needed to wait at least 7 or 8 weeks before I found anything as well. So it can also always change. But after you've waited, then you move to the harvesting phase. You always got to be really careful with the roots because you might be shaking spores, you might be shaking things off the external. Obviously, once it has been infected, all of those structures will be inside the roots, but you also don't want to disturb the mycelium network that is kind of forming on the outside. So it's always a really gentle, time-consuming process. Um, in the clearing phase, the point of this is essentially to get all of the tannins and phenolics out of the roots. So you might find when you pull up plant roots, they could be brown or yellow or different variations of that. And the idea is to try and get all of those colors out of the roots so it's as clear as possible before staining. We do this by firstly dissecting them into smaller pieces. I find it's easier in 1 centimeter length pieces because for my research, I'm actually quantifying the level of colonization. So having consistent sizes is really useful for me. But if you're just having a look, you can just chop it up. And the reason for that is the more surface area it has to get those phenolics to be leached out, it's a lot easier that way. And we're heating in 10% potassium hydroxide. If you do have an opportunity to work with this, you also need to be very careful because potassium hydroxide is a caustic chemical. And if it contacts your tissues, it can cause severe damage like burning or ulcers. So always wear gloves. The optional, once it's heated enough, times can vary. Optionally, you can use bleach to get that final kind of like clearing white look. At the end of the clearing period, your roots kind of look like cooked rice noodles, essentially. You rinse that with water as much as you can to get as much alkaline solution out. Because sometimes, depending on the pH of the roots, some stains might not stick to it very well. So yeah, it depends on the pH of the root, depends on what stain you use as well. But once you've stained, staining time also varies, then you're ready to have a look. Just mount it on the microscope, chuck it down. Let's have a look what they look like. But as you can see already, there are lots of variations, not just in the growing times, but also in the heating times, whether or not you use bleach, which stain you use, and that's found here. So
Vanessa was just asking the temperature for the heating.
yeah, so that can change. A lot of the time it can be basically whether it's so it's bubbling. I used a hot plate and I needed to put the hot plate up to 300 degrees. And although it wasn't actually 300 degrees in the actual bottle, that was just what I had to do to get it to a bubbling temperature. Other methods are using a water bath, or you can put it in an oven and sit it overnight at like 60 degrees. Um, but yeah, you can also use an autoclave, which is kind of like a sterilizing machine that you can find in a lot of labs. So it depends on the plant and depends, like, so for soybean, which has much smaller, more delicate roots, I just needed to boil it at about 90 degrees for about 5 minutes. And that was fine for soybean. But for Pongamia, it's kind of tricky because a big part of my research is that a protocol for Pongamia is not yet established. This protocol is established in some other plants, like soybean included. I know that there's a protocol for eucalyptus, but a lot of the time we're kind of just winging it. We're just guessing and we're just seeing what works and trying different things. So yeah, this is kind of a big part of my research is trying to optimize this protocol to find out how long to clear it for, at what temperature, at what concentration of potassium hydroxide, whether or not we bleach it. And you can use what type of bleach to use, because some people can use standard bleach or hydrogen peroxide. Some people like to acidify the roots before applying the stain, and that's using hydrochloric acid. And again, that's what concentration, how long are we doing that for? And there's also stains. There's a whole bunch of stains we can use. Trypan blue seems to be the standard for AMF visualization, but I've also played with toluidine blue and ink vinegar method. I've used cotton blue in other fungal structures in visualizing even fruiting bodies, like the actual mushrooms that you find. I've used cotton blue to visualize like spores and other things like that. But I'm actually currently testing it how it goes with AMF structures because that needs to penetrate plant roots. So there's lots of different stains that you can use as well. But yeah, you can see this picture here is actually after I've harvested Pongamia, and I had it sitting in the potassium hydroxide for only 2 hours when I, like, just at room temperature as I was preparing other things. And that's how dark the liquid got because of all the tannins and phenolics that you can find in that tree. So you probably have like a common issue with like eucalyptus because eucalyptus will have a lot of tannins in its roots as well. But you wouldn't normally find this with something like soybean because it's already just much lighter and it doesn't have that kind of chemical composition. So yeah, just how many different variations it can have on this protocol. It's a lot to undertake and a lot of things that you can tweak and try and see what works. So yeah, it depends. To answer the question, basically, it really just depends on the plant that you have, and the other factors like concentration. Um, but yeah, depends on the plant. Check existing literature. There might be protocols available for the plant that you're interested in or the plant that you're wanting to check out. But otherwise, it's just kind of like you just play with it and figure out what works over time. And this is kind of what I did for Pongamia. So some of these trials, um, I developed AMF stocks just for the lab so that we can have a supply of different strains of AMF readily available for research. And this was like a really early infection check just to see if the plants were being infected or not. So you can see little bits of hyphae here. That's a arbuscule starting to form, bits of hyphae here. These are little spores. This one's about to germinate. So yeah, this was like super, super early, and this was all in soybean. But then when I was testing the protocol for Pongamia, these were the variations I chose to look into. So clearing time was between 2 hours and 4 hours. Whether or not I bleached it, and I tried toluidine blue and Schaefer blue, which is the ink vinegar as well. So here is the 2 hours of clearing, and here was the 4 hours of clearing. This was no bleach, this was bleach, no bleach, and bleach. And the top row was toluidine blue, and the bottom row was ink vinegar. So you can see small alterations in the protocol can have vastly different results in what the microscopy looks like. So you can see even in here, there are structures here. You can see little shadows, but because of the protocol, you're not able to see them at this. So this one had stain adhesion issues, of course. These should be colored blue, and these structures should be darker. But what I found was that these two are quite similar. So toluidine blue obviously worked a little bit better than Schaefer blue, and 2 hours clearing time with bleach had a pretty similar result to 4 hours of clearing time without bleach. So in terms of efficiency, 2 hours is obviously better, and this was the protocol that I started to work from. So you can see how many different variations, and it's not even testing all the stains. It's not even changing the concentration of the potassium hydroxide. And yeah, there's still so much more we can develop. But at the moment, it's not looking too bad. But for me, the reason I'm trying to optimize this protocol is because I want to quantify the level of AMF colonization in these plants. And there's a couple of different methods you can use for quantification. I use the VBA method because I'm kind of working with earlier stages of infection. There is also the magnified intersections method that is quite standard across the field, but I expect that this is kind of used for more advanced stages of colonization, where you just kind of put it in a petri dish, which has a grid laid into it, and then you view it under a dissecting microscope, and at each grid line intersection, you check whether roots are present, so you have total intersection of roots, and then you count the total intersection of fungal structures, and then you kind of divide the fungal structures over the total, multiply it by 100. That should give you a percentage of infection. So the pros for this is that it's a little bit faster less equipment intensive, and it's kind of suitable for like high throughput studies or large root samples. And you can use kind of more basic microscopes for this. But for me, because I need to look at the roots much more closely on a compound microscope, um, this is the what I use. So I have like a sticker that have all of these lines, only 1 millimeter or 2 millimeters apart, and I kind of just stick it on the back of my microscope slide. And this is kind of what it ends up looking like. So this is just one section under the microscope. So as the root continues, this is one side, two sides, three sides. These outside sections did not have any colonization, but you can see if it's not close enough because I wanted to give you the image of the lines, but these had some arbuscules and hyphae. So essentially, it's just like, okay, well, out of the three sections here, one of them is colonized. So even in just this one picture, you can say it's about 30% colonized. And that's kind of how quantification works. It's nice and simple, this one. I really like it because it's nice to distinguish between different AMF structures. It's quantitative, but it does require access to a compound microscope, which is a lot more expensive and it's a lot more time consuming. Um, and I guess it depends on like the quantitative data can also depend on the scorer if they're used to seeing structures as well, because they might count some things and not count some things. So that's also kind of a bit more dependent as well. Um, but yeah, those are the two main ways we can quantify AMF. Um, but yeah, I guess my just to kind of finish things off, I guess I'll talk to you about what my research is actually working on this year. It's basically you can ignore the jargon, but it's basically broken up into four objectives. The first one here is just comparing AMF colonization in wild type or like normal soybean and common bean plants versus a supernodulating mutant. So, supernodulating mutants have a mutation where they aren't able to regulate how much um, they can't control how much microbes infect them. So they end up having a lot of nodules, and they tend to be a bit stunted because they're using so much energy to develop those nodules. But they're also got a lot of rhizobia. But I'm kind of seeing whether those same pathways that control rhizobia infection also control AMF. Um, the second part is I'm essentially pitting rhizobia and AMF against each other in a kind of a microbial tug of war, and I want to see if they compete or cooperate in wild type soybean. Um, so I'm currently setting this up, and then I'm up to this one. I actually harvested Pongamia yesterday to start looking at this. And I'm essentially wanting to quantify AMF colonization in Pongamia, and I've grown that from seed for about 7 months that I harvested yesterday. So this is essentially going to tell me how effective commercial inoculums are with Pongamia. It's going to tell me how established AMF can be in this species, and it's also able to allow me to test my visualization protocols with more of these plants to harvest and have a look in. The last one is just a bit of genetics, actually. So I'm just going to be looking through the Pongamia genome and comparing it to other legumes to find genes associated with AMF colonization. But yeah, that's kind of this is what I'm aiming to achieve this year. And the visualization protocol is a huge part of that. Um, but yeah, I guess all in all, it's really imperative that we can continue to contribute to this field of research and to mycology, not just for academic research and furthering our knowledge of these incredibly resilient and widespread and intelligent organisms, but to also contribute to the very real world applications of these technologies, and providing nature with its own resources efficiently can do nothing but benefit us in the food security and technology and our own well-being. Yeah, thank you so much for coming and thank you so much for listening. Yeah, I run some plant science events sometimes. I'll sometimes do foraging events with Friends of Nerang National Park. Friends of Parks Queensland do fantastic work for citizen volunteer bush regeneration. So feel free to check them out. They're all over Queensland. Yeah, a lot of my events actually happen at Echo and Bounce, a venue space in Woolloongabba. So feel free to check them out as well. But yeah, thanks for listening.
Thank you, Leisha. Awesome. And just checking if there are any questions. Thank you so much for sharing with us tonight. That was amazing, and love the work that you're doing. That's really exciting stuff.
Awesome. Yeah, thanks so much, everyone.
Yeah, thank you, everyone. Just some comments there. Thanking you very much for your talk.
Thank you, guys. Hope you
Great
liked it.
talk. You are certainly earning your honours. A big project. Wish you success. Well
Thank
presented and
you.
explained. Thank you to the organisers too. Thank you, Epic.
Yeah, thank you to QMS. Now that I've kind of got assessments chilling for a bit, and I'm just focusing on the project at the moment, and I don't have assessments until October. So I'm actually hoping to attend some of the forays now, now that I'm less busy.